Hello everyone, my name is Matthew Crown, and with my poster, I want to present to you a new tool for open and fair metadata annotation in environmental microbiome research, OMEinfo. I'm part of the Microbial Environments Group in the Hub for Biotechnology in the Built Environment at Northumbria University, and I have a particular interest in exploring the ways that our built environment shapes the indoor microbiome, and also our host microbiome. Specific microbiome signatures have been linked to a number of different environmental characteristics, which can have both positive and negative impacts on human health. For example, living in rural areas and with rural microbiomes has been shown to decrease risk of developing asthma in children. And it's also been shown that benefits apply to adults moving to rural locations as well. Small particulate matter and nitrogen dioxide have been shown to negatively impact our skin microbiota, and greenhouse gases, such as carbon dioxide, have been shown to alter the rhizosphere soil microbiome. In addition to this, city locations are classifiable to a high accuracy based purely on their microbiome compositions. It's clear then that global scale environmental characteristics, such as greenhouse gas concentrations, rurality, and location, play a role in shaping the built environment microbiome. But these measures are not necessarily consistent worldwide. Take, for example, rurality. There can be a significant difference between definitions of rurality in systems to measure this, where what one country may describe as urban may be rural in another much larger country. We might think that this is okay, as we can just share the location a sample was taken from, and researchers can use this information to understand relative to their own data what a sample's context is. However, sharing an appropriate level of accuracy for location data and retaining useful information without being so accurate as to identify an individual house, which is personally identifiable information, is difficult, and oftentimes ethical agreements will prohibit sharing this information. At a global scale, there are a variety of different resources available which can richly annotate a location to high precision with measures including the aforementioned rurality, nitrogen dioxide, carbon dioxide, and many, many more through the use of data derived from satellites. For example, Copernicus Sentinel, GOSAT, or AURA from NASA, ESA, and JAXA. This information is frequently used in geographic information systems field. However, it requires a good understanding of the underlying data structures, for example, map projections, scaling factors, raster bands, and software to probe specific pixels in an image, on top of which these data files can be very large, into the order of terabytes in the biggest cases. This makes utilizing such information difficult for microbiome researchers and bioinformaticians without a good knowledge of where to find this data and how to properly use it. So, the problem that we need to find is a way to enable researchers in the life sciences field, including bioinformaticians and wet lab scientists, to be able to easily annotate their location data for samples with a rich set of globally consistent geographical feature annotations in a private and secure way in order to derive new insights from omics data to better understand the role the environment is playing in shaping things like the built environment microbiome. This is where OMEinfo comes in. OMEinfo makes it simple and easy to annotate location data with geographical data from a number of sources, currently covering CO2, NO2, rurality, Köppen-Geiger climate classification, and population density. OMEinfo operates through use of a Dockerized web app, to which a CSV file containing sample metadata, including latitude and longitude, is uploaded. To minimize data transfer and avoid needing to download multiple large GeoTIFF files containing the geographic data, these files are stored in Cloud GeoTIFF format in an AWS bucket. These Cloud GeoTIFF files are queried for a particular tile or subset of the data, which is returned and can be used to obtain information on the specific location requested. In this way, no location data is shared, allowing OMEinfo to be private and secure. The derived geographical information is then served to users through a dashboard, allowing investigation of the data distribution for each feature through both histograms, bar plots, and also on an interactive map. Data can be used and downloaded in a CSV format and utilized in downstream investigations. The underlying datasets used in OMEinfo are maintained in a version dataset, which can be downloaded from the S3 bucket should users wish to make use of this for their own investigations. Finally, users are able to download a bibtext file containing citations for all underlying data sources, and we can also explore the features in a 3D format on a separate page to get a better understanding of what this data might mean to your research. Thank you for listening. And please also try to use the Skull AR app to interact with my poster in augmented reality. Thank you.